I hope we do have a few people that are new that are getting on here that might not have been on Zoom in the past few months, because we've got Lance Wilson tonight, and he's always been a real good, informative speaker. So look forward to him after we have our bee talk. And I do, who is it? Want to bring up the death of Dr. Thomas. A loss to the industry. So remember their family if you can. And we'll just move right on into our bee talk. That way everybody can get more questions answered from Blake Shook. So Blake, you can take it away.
Fantastic, guys. Well, let me share my screen here and see if we can get this to work. It's always an adventure. Great to be back with you guys. So Bee Talk, as you can see on the screen, this is not 2019. I'm hate to break it to you, but this is actually 2020. As much as we probably all love to just go back to 2019, I didn't edit my slide there. But when we think about what's blooming currently, it depends on where you are. There's not a whole lot blooming. The big thing to note this month is that picture in the upper right-hand corner. That's the snow on the mountain or snow on the prairie, depending on exactly what region you hail from. And that just started blooming really last week. It started blooming really well. And it was kind of perfect because we got some really good, 1/4 to 3/4 inch rain last week, depending on where you were. And that was just perfect timing to really kick the snow on the mountain in gear. And some of these sunflowers, the crepe myrtles, are still blooming. And so we've got a bit of a trickle, a little bit of nectar from the snow on the mountain, but we've got a little trickle of pollen coming in, which is wonderful this time of year to have a decent trickle of pollen. Now, what I will say when I'm looking at the weather is this entire week is nearly 100 degrees or more. And that makes me a little nervous because that can really start drying up the pollen and the nectar and what few plants we do have. So decent pollen trickle right now. But again, there's a pretty good chance that by the end of this week, if we don't get some more rains, and it's not really looking like we're going to, that could dry up pretty fast. So kind of stay tuned on that. Some of the current conditions I'm seeing right now is we've had some, I guess, seasonal norms as far as the rainfall. And so the stage is kind of set for a continual trickle of pollen throughout the summer. But again, as you guys all know, being from Texas, I mean, it doesn't take too many days or weeks of 100-degree weather to just really shut everything down and truly go into a pollen dearth. And so, hopefully, if the conditions persist and we get another rain, we'll have a trickle of pollen until late September when we kind of have our main pollen flow begin. As far as nectar, there's little to no nectar coming in. What little we do have is really from that snow on the mountain. And if we don't get some rain pretty quick, that'll dry up. We're kind of having an average summer otherwise, though, as far as temperatures. This week is hot, but it's August, so it's just going to be hot. So anyway, that's kind of my view of the current conditions right now. That being said, we kind of had some nice weeks of cooler weather. Now it's really hot. This is a picture. The picture on the left there is an all plastic frame that I left out in the sun last week. And you can see it has melted every ounce of beeswax off of that frame, and it's horribly warped it. So it's hot. It's hot out there. Don't leave your frames in the sun because it'll do that to them. The picture on the right is where I have a lot of my bees in North Texas. The bees of mine that aren't in North Dakota, but are still here in North Texas. I've tried to get them all into shade, and the picture you can see there is what a lot of my bee yards, my local bee yards, look like right now. I'm trying to get them into shade with at least shade in the afternoons. That way, they're not going to cook out there in heat. A strong hive is going to be able to keep itself cool in general, but it certainly helps if it's in the shade in this, especially August, September, and they're not going to have to forage for quite as much water as well. So this is this next picture. We're doing all of our bee work early in the morning, and it's kind of hard to tell from this picture, but this is before the sun came up. And so when we're doing bee work this time of year, and I really recommend it, whether you have one hive or 1,000 hives, we do our bee work early in the morning or really late in the evening. And not only is that better for the beekeeper, it's better for us because, wow, it sure is cooler. But I think it's better for the bees. When it's 100 degrees outside, you go out there in the middle of the afternoon. If you leave a frame sitting outside the hive for more than a minute or two, it'll start melting that wax. So I try to do the work and recommend doing your bee work early in the morning or late in the evening. Summer care, Texas summers, as I've always said, Texas summers are really tough on bees. It just is. I think the heat is harder on bees than the cold. And so making sure you've got a reliable water source nearby is very important. Make sure that every hive has at least one box above the brood nest. And then ideally recommend cracking that lid for ventilation a little bit. And if possible, get them some shade, at least for afternoon shade. Whether that's moving them into the shade, into shade of a tree, or putting a beach umbrella over them or something to, at least during the afternoons, get them some shade. That could be really helpful and help bring that temperature down in the hive quite a bit. For feeding this month, the main goal is to maintain about 30 pounds of honey or syrup in the second box of the hive. So I'm not talking about just in the bottom box, but in your second box, whether that's a deep or a medium box, maintain about 30 pounds of honey. So that's the medium box, pretty much full, or it's a deep box, about 2/3 full. So you want to try to maintain that 30-pound surplus or so that they don't run out of food. This is the worst possible time of year for bees to run out of food as they're beginning to rear that generation of bees that needs to survive through the entire winter. I would recommend pollen substitute as well. I've been feeding pollen substitute personally since the beginning of July. And so giving a strong hive about 2 1-pound patties per month through November is what I really recommend. And that's just going to help make sure that that the queen can continue laying brood, especially when we have these 100-degree weeks and the pollen sources all dry up. The first thing the bees begin to do when they don't have any nutritional resources coming in is they'll often start cannibalizing that developing brood. And we don't want that to happen, right? And so feeding that pollen substitute can allow that hive to continue rearing brood even when the conditions are not ideal. And then I would recommend trickle feeding syrup rather than just giving them a couple gallons a week all of a sudden until they get that 30 pounds stored up in the second box. I really recommend trickle feeding, which is giving them maybe a fourth or a third of a gallon of syrup per week over a number of months. And that more closely simulates a natural honey flow where they're getting smaller amounts of syrup over a longer period of time. And they're not going to be as likely to fill up the brood nest with syrup. So trickle feeding is a big plus in my mind this time of year. I believe Lance is talking about Varroa, and he does a phenomenal job. I love his Varroa presentation. I always enjoy watching it. And so I'm really not going to talk about Varroa a whole lot. We did touch on Varroa in the July Zoom call. And so suffice to say, the critical levels are peaking now in July and August. We often see those highest Varroa levels, and it's the leading cause of, I put in quotes there, winter death, because so many beekeepers, from hobby beekeepers all the way to commercial beekeepers, if your mite levels get out of control in the summer, it doesn't necessarily immediately kill your hive. It can just really weaken them, and the viruses they pass on really weaken the hive, and then your hive just kind of dwindles in strength over the course of the summer and the fall, and then at some point in the early winter, the hive finally dies. And if you were a couple months late in treating, even if the hive looks okay visually, it could still be too late if the virus load's gotten too high. So I'll certainly let Lance get into all that. The one little note I'll make, which I'm sure Lance will make too, is that a visual inspection of mites, it is virtually pointless. You can see some visual cues that your hive may have a mite problem. I often tell people, if you're to that point, then your hive could be too far gone already. So don't wait until you can see Varroa mice riding around on the backs of bees in the street. At that point, you probably have an astronomically high mite load. So make sure you're testing your Varroa mite levels. Control methods. I'm not going to get into this since you guys are about to hear a lot about Varroa mites. I love what Lance does and how he talks. The one thing I will note is that picture in the upper right there, the Varroa Easy Check. I love that. We use it commercially. We sell it at UBS. It is so easy. I recommend everybody use it. It makes testing for mites so much easier than trying to do a homemade method. So highly recommend that for doing a Varroa test. Okay, I've got a couple random things to just briefly talk about that are applicable to this time of year. And one is just summer hive configuration. That's something I get a lot of folks ask me about and want to know, hey, what should my hive look like this time of year? Should I put all my honey supers back on the hive? Should it be 4 deep stall or a single? And like I mentioned earlier, the thing I really recommend this time of year is just to make sure you do not have a single story brood nest, unless, like I showed you in that picture earlier, it just is in full shade. Otherwise, that sun just beating down on that lid just can really put the hive. So I recommend having at least 2 boxes. Whether that second box is another deep or a medium, either work, but make sure you've at least got a second box on your hive. That's really important to help keep some dead air space between the main brood nest and that lid. And you can keep going higher. What I tell people this time of year is, if the bees are filling up the box, then leave it. If you've got 3 deep boxes, all of them are full of bees, that's okay. I would leave it like that. And then over the fall and winter, they're probably going to shrink back down and eat up some of that honey. You can probably remove a box or so. But in general, as long as the bees are filling the majority of it, I would say over 25% of it or so, then I would leave those boxes on until later in the year. And we'll talk more in the fall about how to pull boxes off, when to do that, all that good stuff. The other thing is empty supers, and this is just a quick side note, and I'm going to try to get through some of these so we can get to your questions, because I see that there are quite a few. Just remember, your empty honey supers really need to be cared for, otherwise the wax moths will feast on them pretty aggressively. So make sure you've got them stored in a freezer or with wax moth crystals. Keep those crystals replenished, and don't store those honey supers in an area that's really, really hot. So don't leave them out in the sun, and make sure that, again, you've got them sealed up and you've got wax moth crystals on them or in a freezer, so that those wax moths don't undo all that work that your bees did. Make sure you've got water sources for your bees, and that's really important. Ideally, the water source is not your neighbor's swimming pool. Make sure you've got, if you're in a neighborhood, then make sure you're providing a water source for them. If you're out in the country, make sure you've got a pond or a creek that doesn't go dry within a quarter mile or so. One little note, I know that bees in swimming pools, neighbor swimming pools, are a major issue. One little side note is, if your hive is shaded, again, they won't need quite as much water, so that might help you out a little bit. in the neighborhoods. Peppermint, somebody told me recently that peppermint oil can help discourage bees. I was talking to someone that sprayed it around the perimeter of the school, and that helps discourage bees as well. So, quick note on, and I know this slide is still loading up here. There we go. But bearding and flight, you're definitely going to see a reduction in bee flight in these hot afternoons, from noon to 5 or 6 o'clock. Bees probably aren't going to be flying a whole lot. You might see some bearding, like you see in this picture, especially in the evenings if all those foragers come back and it's really hot in the hive and they just kind of hang out on the front. It's not necessarily a problem. It doesn't. However, if I'm seeing bearding like this picture, I like to add another box or provide them some partial shade. It's natural for a really strong hive to beard out like that on hot days. But it does tell me that there's not quite enough room in the hive and they're struggling to keep it cool. So, if I'm seeing this on my hives, I do like to take some form of action. It's not critical, but, give them an upper entrance to ventilate, give them another box, give them some afternoon shade, something to pull that bearding down just a little bit. Okay, I think this is the last slide, and we can probably email this out if needed, just because it's kind of hard to read on this screen. Depending, if you're viewing it on a computer, you can probably see it fine. If you're viewing this on a tablet or smartphone, you can actually zoom up right now and see it. But this is something we put together recently that I think is a little bit helpful to briefly address this time of year, because a lot of people have weak hives, really any time of year, but especially this time of year. And this is a tool that might be helpful in diagnosing why hives are weak and what to do about it. And so if you've got a weak hive this time of year, or the reason it's important to talk about it in August is you might still be able to save it. If it's under 2 to 3 frames of bees, it can be tough to save a hive that week. But if it's over, if it's 3 really good frames of bees or above, then you may be able to save it. We've got about 2 to 2.5 months left of potential growth season, if you will, for bees. And then it just, mid-October or so, it just starts getting too chilly and the bees really shut down hard. But if you've got a weak hive now, you've still got some time to do something about it to try to get it strong. And so these are 6 of the most common things I found that cause weak hives. There are certainly caveats. There are certainly other things that cause weak hives. And I'm just going to run over these really quickly with you. And feel free to reach out, or we can email this out. And if you want to take a look at it closer. But one of the leading causes of weakened hives is Varroa mites. And so ask yourself, if you've got a weak hive, are your mite levels under 2 per 100 bees? If not, then do a treatment and then test afterwards and make sure it works. Does your hive have at least 3 to 4 full frames of honey? And I'm going to say that is at a bare minimum. If not, then feed pretty aggressively to boost up their food stores. Does your hive have half a frame plus of stored pollen with fresh pollen coming in? If the answer is no, if you're not able to see hardly any stored pollen in the hive, or if you're seeing pollen that is only one color, then I would definitely recommend feeding pollen substitute. Just as a general rule of thumb with pollen substitute, don't feed more than a hive can eat in about a week. You don't want the small hive beetle larva to hatch out and really start aggressively taking over the hive and making a mess. So I usually give about what I'm guessing a hive will eat in a week. And then at the end of that week, if there's still some patty, then I'll take it, throw it in the freezer for a day to kill any small hive beetle larva, and then put it back in the hive. Number 4 is the brood pattern spotty on every frame in the hive? If the answer is yes, then if your mites are under control and sufficient food is present and your brood is still spotty, good chance you need to re-queen that hive. Number 5, is there any room for your queen to lay, and are there fresh eggs and larvae? If there's no room, if every frame is full of nectar or full of honey, then stop feeding or add another box to give them a little bit of room to they may be honey bound. If there's no eggs or larvae, check and see if you've got queen cells. You may need a new queen. And then finally, does the developing brood look pearly white and healthy? So when that brood is in the larval stage, you know, is it pearly white? Does it look healthy? Is there a lot of larvae? Or is that larva kind of twisted or brown or yellow? Does it look unhealthy? And if that's the case, you might need what I often recommend is probiotics. You may have European foul brood, you may have hog brood, and I try to use probiotics instead of antibiotics where possible to try to clean up some of that dirty brood, if you will. Okay, so as always, I ran through that pretty aggressively. I know we're always short on time. I'm going to stop sharing my screen here and go to your questions. Let me see. There we go. There's that button. Okay, there we go. All right. And let me see if I can find your questions here. There we go. And Russell, how am I doing on time? You're muted there, Russell. I think you said you're fine, though. I was trying to read your lips there.
Yeah, about 10, 15 minutes. We should be
okay. Awesome. Okay. Well, I'll try to run through these questions. I want to make sure that Lance has more than enough time. Okay. Scott, you ask, for a hobbyist that doesn't often move hives, is it worth moving them to shade for one hot week? We don't have a yard 3 miles away to move them to. So, no. You know, I wouldn't recommend moving them for one hot week. My recommendation is usually to provide some shade starting in, you know, early July, you know, late June, early July, providing them some shade all the way through September. So, you know, for one hot week, no, I don't think that's worthwhile. But, you know, if you can provide them some shade through the entire course of the summer, I do think that's worthwhile. Someone asked, doesn't shade also provide ideal conditions for bee pests to thrive? So there's two things I worry about with shade and pests. One, the biggest one is small hive beetles. Some brood diseases can thrive a little more in cooler, wetter conditions. I don't worry about that much in the summer. It's neither cool nor wet in Texas in the summer. So the only thing I'm really worried about in the summer is small hive beetles. And again, a good strong hive that is able to typically take care of those and keep them under control. If you're concerned, you can put a small hive beetle trap in the hive and just make sure you don't have syrup or pollen patties sitting in the hive more than a week or so, because that's really where I see small hive beetles aggressively start taking over the hive. But in general, at least some partial shade in the afternoon isn't going to make small hive beetles take over your hive, and it's worth it. And I've been meaning to do a study on this and do some personal research on this for a couple of years, and maybe someone else has done it. But I'm really curious to know if small hive beetles are worse in the shade, or if they just tend to be up on those top bars more in the shade because it's cooler. And so when you open the lid, you see small hive beetles all over the top bars, and it appears that they're worse. Versus in the sun, or they just on the bottom board and in lower, cooler areas of the hive. So someone may be able to educate me on that. There may be a study out there that shows, are they actually worse in the shade, or do they just appear worse? So I don't know. Someone asked, is there a proper way to move bees to a new area within a yard so we don't confuse them? Great question. So the best way to do it is just to move them a couple of feet per day. And you don't want to move them, you know, 50 feet all in one day because you'll confuse them. But if you move them, you know, 5, 6, 7 feet per day, then they'll kind of move along with that hive. Have you heard about, someone asked about for the mite test. Actually, a couple people asked this. So when you do a Varroa mite test, the normal recommendation is to use rubbing alcohol. So you put the bees in that cup, you pour, you know, about a cup to, Lance can probably tell you better than I can, but there's on the easy mite test, there's a fill level where you put bees in at a certain fill level, and then you pour a couple of cups of rubbing alcohol in. and it kills the bees, unfortunately. But then you can see the mites kind of floating in that rubbing alcohol in the bottom of the jar. You can get a really accurate count. As you guys, I'm sure, are aware, and I've come across this problem too, you can't find rubbing alcohol anywhere. No one's selling it right now because of COVID. And so Russell was asking it, have you heard of using Dawn dish soap instead of alcohol? And yeah, we've started using that. Randy Oliver is suggesting that, and I found it to be every bit as good as the rubbing alcohol. And so I can't remember the mixture. I think it's 1 or 2 tablespoons of Dawn dishwashing soap to a gallon. I think I see Russell and Lance nodding, to a gallon of water, and use that in place of alcohol. It seems to work really well. I've also used vinegar lately, and I've gotten equivalent counts of vinegar. So yeah, recommend that. Someone asked, is a 6-step for diagnosing weak hives available as a downloadable handout? If so, yes. Yeah, I can send that to CCHBA, and then you guys can definitely send that out to whoever you'd like to. Someone's typing, so it's 1 tablespoon of dish soap to 1 gallon of water. So yeah, that can be used in place of rubbing alcohol. Someone asked, when is the best time to replace old frames with new frames? So, I try to rotate my frames about every 8 to 10 years. I try to get rid of all the old foam and replace it with new foam. And I just do that whenever that old foam becomes empty. I don't want to throw away a frame full of honey or a frame full of brood. And so usually that's over the winter when I have a hive die, or if I have empty frames on the outside, that's when I like to replace them rather than when they're full of honey or brood. Douglas asked, why test for mites? Why not go ahead and treat? Is it to compare before and after? So not every hive has a problem. You may have a hive that or a couple hives that don't need to be treated, and mite treatments come with some detrimental side effects. And but so I wouldn't test unless there is a problem. Most people have a problem. I mean, mites are in most hives, but I'm going to let Lance get into that a little deeper because he's very, very knowledgeable about it. Sandra asked, how much vinegar? So usually, I'm guessing here, I'm going to say about 1.5 cups of vinegar, or 1.5 cups of that dishwashing soap solution. It's about what you need in those test jars. It doesn't have to be exact. You just want enough to soak the bees and rinse. You're essentially rinsing those mites out of the bees and then counting the mites on the bottom of that test jar. Okay, I think this is the last question. For a honey-bound hive, oh, there's another one. For a honey-bound hive, can I just stop feeding sugar water for several weeks during the dearth to force them to use stored honey and open that frames up? Or should I remove honey frames and replace with new foundation? So if you've got a honey-bound hive, I would say it depends on how badly honey-bound they are. So if they are to the point where, if they're down to 1 or 2 frames of brood and that's it, and everything else is completely full of honey, then what I would do this time of year is, number one, I would certainly stop feeding them. I would take a frame full of honey right next to the brood nest out, let the bees rob it out, empty it, and then I would replace it right next to the brood nest to give that queen a fully wide open frame, or 2 really, to start laying on again. I would try to replace it with a robbed out frame rather than foundation, so that queen can start laying again pretty aggressively. So you can also add a box onto your hive and let them hopefully start drawing out some foundation and moving some of that honey up and out of that brood nest area. Okay, I think this is the last question unless one more pops up. After 1 week, my pollen patties dried out and are rock hard. What can I use to keep them moist longer, and do I throw away the hard patties? So there's a couple things there. Pollen patties getting hard. Two main reasons I see that happening. Number one is if you mix them up yourself. A lot of people will use sugar water to mix them up, and that guarantees they're going to dry out incredibly quickly. If you mix them up yourself, you've got to use corn syrup to mix them up, because that retains its moisture and keep them from drying out. The next thing is, if you're finding they're drying out quickly, if you pull the wax paper off both sides, that also dries them out quickly. So I recommend leaving the wax paper on both sides. It's made really thin so the bees can easily chew through it. That wax paper is there to help retain the moisture in that patty, so it doesn't get hard. The other thing is that if the patty started out hard, it's kind of a lost cause. The patties, when you get them, should be able to compress them with your fingers. They should be decently soft. Once they get hard, I usually throw them away. I guess you could try soaking them in a little bit of water, and dunking them in water and put them in a Ziploc bag overnight and try to get them to rehydrate. I feel like I just kind of make a mess when I do that, but you could try. I know some people have thrown them in the freezer and then pull them out and let the condensation kind of moisten them again. So you could try some stuff like that. You just want to make sure they don't get moldy, and attempt to kind of rehydrate them that way. But in general, if they're hard, I usually throw them out and start over and just make sure that wax paper stays on them. And then the other reason the other reason they can get dried out is if they're just in the hive too long. If a patty's in the hive, this time of year especially, for 2 to 3 weeks, it can certainly dry out. And so if you've got a hive that is weaker, then I would just give them maybe a third of a patty at a time or a half a patty at a time, something that they can consume in a week to 10 days. And that way the beetles won't be a problem and it won't dry out. So hopefully that answers it for you there. Okay, that is all the questions that I see. So if there are no more, then Russell, I will give it back to you.
I've got a bottle of alcohol if you want to trade like maybe 20 queens for.
Hey, at this point, it's probably worth that. You could probably sell it on eBay.
Okay. All right. Thanks, Blake. Really do appreciate it. Okay. All right. Next, we have Lance Wilson, and he will be talking about mite treatment with the next portion of the program. Go ahead, Lance.
Okay, so does everybody see my app? See my screen?
I see your screen.
Okay, good. All right, well, I'll start by saying thank you so much for inviting me. I really do appreciate it. Originally, when we made these plans a long time ago, before the virus, I was, of course, planning on going there personally, so I'm so sorry I'm not able to see you. But thank God for technology. And I'll tell you something interesting. I heard on the news recently about Zoom, this technology that we're currently using, that this particular company now is worth more than the Hilton Empire. All the hotels, Hilton Empire, all the hotels all throughout the world, this program is capitalized at a higher rate. It's just amazing. But anyway, we're here to talk about Varroa mites. And we're going to talk about Varroa mites in the context of integrated pest management. So let's go ahead and get started. So the first thing you need to know is really the Varroa mite is really public enemy number one. This is the biggest problem that we have in beekeeping. It arrived in the United States in 1987, and then immediately it just killed 90 to 95% of the feral populations. Now, apparently the data that they have is from a study in Southern California and a study in Northern California. And in the Varroa mite came in 87, and it had been distributed largely throughout the United States in the early 90s. And so this Southern California study was done in let's see, I think it was 92, if I remember correctly. And so in 1992, when the study was done, they had lost almost all of their feral populations, but they had previously surveyed the feral colonies before the Varroa mite introduction, and they realized it just wiped out the population. And this is really kind of interesting because so much, unfortunately, of what we're learning about biology because of the virus teaches us something about beekeeping and about this host parasite equilibrium. By that I mean when a novel virus or a novel pathogen is introduced into a population. In other words, a brand new pathogen is introduced to a population that biologically has no biological experience with it. It just kills, wipes out the most susceptible group in that population. And in this case, of course, it was just a huge percentage of the honeybees. And in fact, there was a study that was done in Canada in 2010. And what they did was they basically did a survey. And what they were able to come up with is that apparently the Varroa mite was either directly or indirectly responsible for 85% of all the colony collapses. I think it was in 2009. So you can see this is a huge problem, and this is all about a naive or new pathogen being introduced to a susceptible population because there's been no equilibrium that's been created. You know, typically what happens is that a novel pathogen is introduced into a population, a huge swath of the population is affected or even perhaps even killed, and then the more tolerant population survives and it reproduces, and so it reproduces this tolerance. And the pathogen doesn't really want to kill its host completely off, right? It just wants to reproduce, because if it kills off its host completely, then it results in not being able to reproduce. And so the host, there's selective pressure for the host to be able to build a tolerance. And then in addition to that, the pest or pathogen creates, there's selection behaviors to select those particular pathogens that are engaging in behaviors that make it so they're not nearly as harmful or virulent to the host population. And so by that, eventually there's an equilibrium where they both live side by side in harmony without any real significant problems. But that's not where we're at now. Where we are now is that this is a new pathogen that was introduced, especially back here, in 1992. And so all of this biology has resulted in a huge sea change in what we do as beekeepers. And in fact, if you're going to be a long-term beekeeper, you're just going to have to incorporate practices that results in keeping your pest population and your parasite population in check, or you're just not going to be a beekeeper for very long. This was kind of an interesting graph that I found. And what you can see over here on this axis on the left is this is the number of managed colonies in the United States in millions. And you can see in World War II, then World War II, there was just a huge number of managed colonies. They needed the waxed waterproof canvas and all sorts of military uses. And you can see that it's gradually decreased as time has gone on. But you can see where the circle is there's this precipitous decrease. And this is where the Varroa mite had hit. Actually, to be perfectly honest, it's when the trachea mite and the Varroa mite hit. So it was kind of a double whammy. But it resulted in just a lot of beekeepers just got out of the business. And if they were just hobbyists, they just thought it was too much trouble to manage mites, even if they were able. And so they just got out. And this is the reason why. It's a host jump that took place in 1957 in regards to the Varroa mite that was living on this bee that you see up here, Apis cerana. And Apis cerana is referred to as the Asian honeybee. And it's the closest cousin really to Apis mellifera, which is our western honeybee. In fact, it's thought that Apis mellifera, our western honeybee, radiated from, in an evolutionary sort of way, directly from Apis cerana. And so that's why the Apis cerana always looks, really very similar to Apis mellifera. It's just a little bit smaller. But in any case, the Varroa mite's been living with Apis cerana for we don't know how long, but for a very long period of time. And because of this really long-term relationship between the mite and the virus and the bee, it's resulted in this equilibrium that I referred to, where the bee is able to accommodate a certain number of Varroa mites without any dire consequences whatsoever. And the bee has incorporated behaviors that's resulted in keeping the mite population low. So they have found this equilibrium where the mite still is able to reproduce while at the same time the bee's not having any problem and really prospering. And so you see a map here of the United States, and in red is the known area of occupation of the Varroa mite. And of course, it's all throughout all of Africa as well, even though it's not indicated here, but it's very difficult to get surveys, information out of Africa. And you can see that it's not yet in certain other parts of the world. But it's getting there. It'll be everywhere eventually. Australia, they're just shaking in their boots. They still haven't had them, but they expect to get them eventually. And a lot of like New Zealand, for example, which is really close, Varroa mites arrived there in the year 2000. Now, so what happened here was you had the Apis cerana, this bee, this Asian honeybee, that had the Varroa mite. In the middle of the 1800s, there was a Trans-Siberian, let me see if I can get my pointer out. There's a Trans-Siberian rail yard that was made all the way from Ukraine and was built all the way close to the Koreas. Can you guys see my mark here, my marker, my cursor, my pin? Anybody?
I see it. Yeah.
Okay, good. Okay, it's working. That's good. Okay, so in the middle of the 1800s, the Tsar builds this crazy long rail line, right? And some of the beekeepers that were keeping our Western honeybee in the area close to Ukraine jumped on that train with their colonies, and they moved them all the way over here to the other side of the continent and started keeping bees. And what happened was that the Varroa mite then jumped from this host to our Western honeybee, and this is referred to as a host jump. Now the problem is that our honeybee had none of the selected behaviors that has been protecting Apis cerana for all these years. And so as a consequence, that was why we've had such a precipitous drop in the number of honeybees in the United States, as they're coming to this brand new virus, this brand new vector of Varroa mite that transmits these viruses. So I'm going to go ahead and show you the latest in microscopy so that for the first time, really, you can see what a Varroa mite looks like really close up and personal. And apparently they look exactly like this. So you probably guessed they don't look exactly like that. But the reason why I show you this slide is that effectively that is really what a Varroa mite is, right? It's just a little vampire. But mites more specifically are really quite interesting. There's about 50,000 described mite species in the world, in other words, mites that we know about, and they suspect that's maybe 5% of the population in terms of species, that there's another million species that's unidentified out there because mites are so small, but they have certain things that are all in common. First of all, they're blind, and they really understand their environment specifically just through smell, taste, and feel. They have a wax coating. Now this wax coating, these have a wax coating too. The wax coating, in case of the mite, it allows the mite's wax coating, because it's lipophilic, to capture the smell of the colony, which helps it move around and not be identified because it can't be smelled by the bees. And you've got to remember, bees are living and evolved in dark spaces, right? In cavities, in a tree, and of course we move them into our hives. And so they really understand the world around them when they're in their colony, almost exclusively like the mite, through the sense of smell and taste and feel. Now, it's an ectoparasite obligate, which is a real fancy word, but as an ectoparasite, all that it means is that the Varroa mite is on the outside of the bee and feeds from the outside. And it's an obligate, and that's kind of a cool biological word for a beekeeper to know, because an obligate means that the parasite is obligated to the host in order to complete its life cycle. So if you understand that the Varroa mite is an obligate, then you know that if you have a colony collapse from Varroa mites, you just need to shake out the equipment and you can reuse it. It's not like a brood disease, it should be fine. So in order to be able to understand the big picture of how to manage your mite in terms of timing of abatements, you need to understand there are two stages so that you can kind of know how to hit them properly with whatever it is that you're wanting to do. And the first stage is called the reproductive stage. So what happens here is that in Apis mellifera, the worker, they refer to it as the post-capping period. It's about 12 days, right? So bees, just to back up a little bit, they basically mature using multiple stages. They've got complete metamorphosis going on. So the first stage, of course, is an egg. An egg is about 3 days, and then you've got a larva. The larva usually develops over 5 or 6 days. And then what happens is that the cell is capped by the bees, and this is referred to as the post-capping period. It's about 12 days, and this is where really all the metamorphosis goes on, where the grub or the worm, the larva, it transforms itself into a bee. And so after 12 days, the bee escapes from the cell. Well, what happens is that the foundress mite, which is a mated female, and you can see her right here. Remember, she understands her world almost like bees, exclusively through the sense of smell, touch, and taste. And so the foundress mite will smell the odor that's given off by the skin of the larvae to know to jump in right before capping. The mite times when she jumps in, because if she jumps in too early, the bees can identify her, grab her, and pull her out. But instead, the mite then, there's been selection pressure over many, many years to time it, so she just jumps in right before she knows the bees are going to cap over that cell. And so this foundress mite will move low, typically hide underneath the feeding jelly that's down here, typically at the bottom. And after the cell is capped, she'll move up and she'll typically feed on the larva. First, after 60 hours, she'll lay the first egg, which will be male, and every 30 hours after that, she'll lay a female egg. And so after the course of 12 days, on average, about 1 to 2 mated females escape, along with the foundress mite. Now, this reproduction rate might, on the face of it, not seem like it's very robust, but mathematically, the population dynamics of this kind of reproduction for the Varroa mite means that she can double her population every 30 days. That's exponential growth. And then again, going back to the virus, you've seen all those exponential graphs, which are all very similar, and then they start with a very low gradient, and then they just automatically, they start shooting straight up. And that's the very nature of exponential growth, the very nature of something that doubles, for just a short period of time. And the same thing happens with the mite. It just stays at what appears to be a low level and just kind of takes off in a colony. The other stage, the second stage, is referred to as the dispersal stage. They used to call it the phoretic stage, but they're now calling it the dispersal stage. And you can see a mite right here on the bee. Now, so after the foundress mite leaves with 1 or 2 mated females, they'll make their way and they'll typically find a bee that's flying in order to be able to disperse to other colonies. Now, what I'm going to go ahead and make a point here is that you'll typically not see a mite on a bee like this. Typically what happens is that a mite, when she feeds on the bee, she'll move to the underbelly of the bee, and there's intersegmental, or there's exoskeletal plates. They refer to them as scorites. And these plates, if you can see, I'm kind of trying to just display what it is I'm trying to show you here with my hands, but you see that basically the plates will overlap, but they're attached to an intersegmental membrane, which is elastic, which allows the abdomen to expand, for nectar collection and breathing and that sort of thing. Well, the reason I'm telling you all of this is because the mite's able to move in between these 2 plates underneath the belly and virtually hide itself almost entirely from you being able to see it when she's feeding on a bee, which is almost all the time in the dispersal stage. So when Blake tells you that visual inspection is virtually pointless, he's only telling you that because visual inspection is virtually just pointless, because you're just not going to really see mites on bees usually, and you can have this huge infection rates of mites and just not really likely to see one. There was one bee specialist, I remember in a lecture one time, he says, if you're waiting to visually inspect your bees, to do something about your Varroa load until you first see your first mite on a bee, then you're just basically counting the colonies that aren't going to make it through the winter, because it doesn't matter what you do, at that point it's too late. And the reason why it's too late is because of this, and this kind of explains a lot of the population dynamics that goes on. But you can see on this axis, you have the number of bees all the way up to 40,000. And then you have the months of the year down here on the bottom. And of course, early on, like in January, the mite population, well, the bee and mite population started out really quite low. And what you now know about mites and how they have to have a brooding area in order to reproduce, because the founder's mite's got to jump into an open cell. Because of that, you know that a mite's ability to reproduce and grow in your colony is going to be directly proportional to how fast your colony is growing or how much the brood area is growing. And so you can see that, bees are brooding up, and so they've got a big brood area, and so they're growing at a pretty high clip here, and your mite population is going at a pretty good clip too. In fact, you're doubling. This is a doubling. This is what it looks like, double every month. And like I said, exponential curves are all the same. All of a sudden, they just take off and go virtually straight up. Now, if you go and you look at your bees, looking to see if you can see mites, say in January, February, or March, you're likely just not going to see any. And it even becomes worse later on in May and June, because even though your mite population has been doubling, you've got so many bees in your box that by visual inspection, just by happenstance to see a mite on a bee would just not be very common. And so the problem is that the time that we've got going on right now is typically a pollen dearth. And by that I mean there's a segment in the middle of the year where there's not much pollen going in. And as a result, the bees are environmentally adapted to their environment, very responsive. So when their pollen decreases, they decrease their egg laying and their brood area because they don't want to have so many bees or so many babies that they can't feed. That would be a terrible waste of resources, right? And bees are nothing if they're not frugal and economic. And so anyway, so their bee population, because there's not any pollen coming in, it's decreasing, but because there's still so much brood in the box, you might still have plenty of opportunity to reproduce. And so look, you just got this, mite population is taking off compared to the bee population. And at this point, maybe in August, you've got so many mites relative to the number of bees, maybe you really do see a mite on a bee, and you go, oh my gosh, I got mites. And I'm here to tell you that no, man, you've had mites this whole year. Your mite population has been careening out of control every month. It's just that it's completely invisible to you because you're relying on visual inspection. And what makes matters worse is that all these mites are injecting a viral load into the bees. And so if you were to, like in August, if you were to wave a magic wand and kill every single mite in your box, it wouldn't make a bit of difference because so many bees have been bitten by so many mites and injected with such a significant viral load that the viral load is its own dynamic. It's its own animal. It's now often its own, it's doing its own thing, killing your bees, and there's no way to put that genie back in the bottle because you didn't control your mite population early. And the reason why it's so critical here is you can see this, that says winter bees up here. And what this means is that these bees that are made in the winter are physiologically different than the bees that are made in the summer and the spring. The bees in the winter are made with huge fat protein bodies called vitellogenin, which provides an immune response to diseases, but in addition to that, it allows bees to live for a very long time. It's called the fountain of youth. And but anyway, what's happened here is that you've bitten all these winter bees that have got to last the winter that are loaded down with these fat bodies, and the viruses are likely to kill them. They're likely to kill those winter bees. And that's why you've got so many dead outs in the winter from people that aren't monitoring their mite loads, earlier in the year, because you've got to have clean winter bees if you're going to make it through the winter. And if your winter bees have been bitten by Varroa mites because you didn't properly contain them before the winter bees were made, then you could be in some trouble. Now, the mechanism of all this is kind of interesting to me. It's not really the Varroa mite. I mean, the Varroa mite, yes, it causes mechanical damage to the bee, to be sure. It's not a good thing. But it's the actual inoculation of a viral load into your bee from the Varroa mite that's really what's cratering our colonies. And what makes matters worse is that the Varroa mite is selecting not only for a large volume of viral load, but it's also selecting for the viral types that are the most damaging to bee colonies. And there's been several studies that have indicated that that really these Varroa mites are just really terrible, just because they're able to protect the viral load and really select for the viral loads that are going to be really the most damaging. So that's why I put kind of the barn with the barn doors open. You know, if you basically, if you're not watching your viral loads and doing something, it's by August it's too late. You know, the horse is already out of the barn. It's too late to shut the doors. And it's all because of this viral load. The Varroa mite is just a vector for the virus. You know, it's what actually gets the virus to the bees. And that's what makes it problematic. The problem is we don't have treatments or abatements yet for viruses. So really the only way to control the viruses is to control the vector, that being the Varroa mite. Just in the same way as that we control malaria by controlling the vector, the mosquito. And I'm going to go into the weeds just a little bit more, and then we'll back out and talk about practical stuff. But I'm going to tell you this because I always thought this was kind of interesting. It's the reason why the viral load and the Varroa mite are so damaging to bees is because the Varroa mite has allowed a mode of transmission shift of the virus into the bee. By that I mean that these viruses have been around forever, since the beginning of time. It's thought to be around since the beginning that the bees started evolving. And the bees have lived with these different sorts of viruses without any sort of problem. So why now are viruses killing colonies? And the reason is because the Varroa mite as a vector is able to create a mode of transmission shift in the way in which the virus gets into the bee. Historically, for millions of years, the viruses will come in orally, and there's all sorts of mechanisms in the microbiome in the midgut that help protect against diseases. And these mechanisms evolved over millions of years just for that very reason, to contain viruses. Now what's happened is because the mite bites and basically injects the viral load directly into the bloodstream of the bee, it bypasses the midgut and all the mechanisms to control the viral load. And so that's really the big problem with the Varroa mite, is the way that it's transmitting these viruses for the first time, which makes it really quite unique. So what are some of the symptoms of collapse? Well, the first thing you're likely to note is, if you see over here on the left, there is really spotty brood. And this is really bad brood, right? And you can see that there's some death going on in here, and they're opening up the cells to kind of eliminate the various larvae, pupa that have died. And this is kind of what you see. And what you're seeing here is that the viruses are interrupting the immature stages of development of the honeybee. In other words, it's killing the larvae, and it's killing the prepupa, and it's killing the pupa. In fact, what's uniquely diagnostic of parasitic mite syndrome, which is what this is referred to, is that it interrupts every stage of or kills every stage of the developmental process. This is the only brood disease that does that. All the other brood diseases will interrupt the immature developmental process at one particular point in development, whereas viruses don't. They kill at every stage of development. And so that's what you use as a beekeeper to decide that you have, say, parasitic mite syndrome instead of, say, American foul brood or European foul brood or chalk brood or something like that, is you kind of look at what stage of development is it interrupting. And if you look and you see that, you know, you've got a young larvae dying, and then somewhere else in the front there's an older larvae dying, and you see a prepupa dying, that kind of thing, you're seeing different stages of death at different ages, then these are viral loads. This is viral death. Now, you only see the brood death at very high concentrations of viral loads. And then the other thing that you'll see in regards to the adult bees, you see these crinkled wings. The deformed wing virus are responsible for this. the deformed wing virus, once you start seeing bees that look like this, typically you'll see them, if you haven't been monitoring or controlling your mite loads, you'll start seeing this typically in August now, in September, but you could see it earlier. But whenever you see this, this only occurs or manifests itself at huge concentration of viral loads of this particular virus. In other words, once you see this, man, you're in big trouble, you know. Your viral loads are probably too high to even be contained at this point. So I don't know if you can see this, but I've got this little red circle that's that I've kind of, you know, marked all my Varroa mites here. And there's Varroa mites everywhere on these bees. This is on the telltale or the end of the collapse of a colony. And so you can see all of these, the virus or all these Varroa mites were just left on the remaining bees. Every one of these bees was dead 3 days later, to give you some kind of idea. This is the tail end of the collapse, as I was telling you. And that is a very critical characteristic of Varroa mites in terms of understanding the dynamics of Varroa virus collapse. There's been so many beekeepers that have come to me that will have a colony not quite this high. This colony must have come out of Canada or something. I don't think we get, well, maybe Blake does. But anyway, what happens is you got a colony that's really just full of honey and bees, and beekeepers, you know, they're so happy, all's right with the world, and they come back and 2 weeks later, man, and there's not a bee in their box. The colony is just gone. And what they'll tell me is, usually it's like, hey, I must have Africanized bees, my colony absconded. And really, that's not, chances of that happening, that's not really what happened. I mean, the chances are that it was just Varroa mite collapse. And that's the nature of behavior of what happens with social insects. There's a biological term called apoptosis. And what apoptosis is, it's a word that describes a situation in social insects where if one insect gets sick, if it's living in a social environment, then it will display behaviors of social apoptosis. And by that I mean, if it gets sick, it will leave the colony, because you need to think of a colony as not of each individual bee in a collective, but more as the colony of bees being a social organism. And it's really a superorganism. And as a result of that, it behaves like a superorganism in that if any one bee, as being a cell of the organism, gets sick, it flies away, because its main concern, of course, is the mother ship, the superorganism. And so it's just a way socially for animals to kind of help maintain, you know, their mother ship, as I put it. Termites do it too. I mean, termites, they're social insects that live in these big mounds. And what they'll do is when a termite gets sick, it'll stamp its foot on the ground, and all the other termites know to stay away from it until it can make its way out and walk in and die away from the mount. And so bees do the same thing. And so when a beekeeper is saying, oh, my bees absconded, no, social apoptosis, baby. You just didn't know that your colony was collapsing through Varroa mites, and you came back 2 weeks later, and all your bees had made individual independent decisions because they were individually sick to leave the colony, which resulted in rapid depopulation with just in a couple of weeks. And then one of the other signs that you can see after you open up a colony and all your bees are gone, is sometimes you'll see this, you'll see the bees as that they were trying to leave their cell, trying to emerge. They had such high viral loads that they just died in their cell. This is uniquely diagnostic of this particular disease. You don't see this anywhere else. And so this is kind of what happens, right? You've got a bunch of colonies here, and you've got one colony, and its mite load just takes off and kills the colony. That colony depopulates, and so now there's no guard bees that are defending the honey stores. And guess what happens? All the other bees go over to grab that honey, and all of those, remember that slide that I showed you with all the bees, with all the Varroa mites? Well, that's what's hanging out in those dying colonies. And when other robber bees come in from other colonies, your colonies in your apiary, they're going in to get that honey. All of those Varroa mites on the outside of those dying bees jump ship. They're like rats leaving a sinking ship. They will jump on to this new bee from another colony, and then those mites then will be spread throughout or dispersed throughout your colony at really high levels. They refer to this as immigration. And they've noted that you can get hundreds of mite increases in a day on the basis of this immigration. So it just kind of really blows up what it is you're trying to do in terms of keeping your mite loads down. So this brings us to hopefully maybe a source of tools that help us deal effectively with this problem that's referred to as integrated pest management. Now, integrated pest management was not invented on the basis of beekeeping. Integrated pest management is a system that's used in all sorts of, almost since the 1950s. Since there was an explosion of pesticide use in the 1950s, integrated pest management was kind of the answer to that. And the whole point of integrated pest management is to employ all sorts of different types of behaviors and abatements. And any one management system that you use or abatement or something that you use to reduce your mite load, any one of which might not be enough to keep your mites under a manageable level. But the idea is that you're attacking the mite from various angles. And so that although any one behavior won't be enough to keep the mites under control, the argument is that collectively, every single abatement you're using is taking a little bit of a bite out of the mite population, that collectively they will keep you under a threshold that will cause disease. And that's kind of an important thing conceptually for you guys to understand. In biology, all organisms have parasites, and they are able to come up with an equilibrium, so they're able to house and host these pesticides, or I'm sorry, parasites without much ill effect. Because they've had a time to create this equilibrium. But the thing is that you've got to get there, right? And so basically what we're doing is we're going to employ all sorts of different things to try to help us maintain this equilibrium so that the mites don't get to such a high level relative to the bee population. And so we start with culture and we move all the way up. So let's go ahead and talk about that now. First thing that you need to understand is that you've got to monitor your mite levels, right? Because if you don't know what your mite levels are, you don't really need to, you don't know if you need to put in a treatment or not, or if there's something else that you need to be engaging in order to try to keep those mite levels under control. So you're going to have to monitor your mite level, as Blake's already made the point. Visual inspection for mites is just pointless. The other thing you need to understand is that if one colony is above threshold, in other words, if you've got more than 2 mites for every 100 bees in your colony, that's referred to as action threshold, and you need to try to do something in order to try to move your mite population down. But the point I was trying to make is that if you find one colony that's over threshold, then what they're telling us now is that you need to treat all your colonies, because of the dispersal nature of the mite is so robust that the only way really to keep them under control is to really treat every single colony in your apiary, even if you only have one that's over threshold. The other thing I like to point out at this point is that after you treat a colony, you need to retreat it a couple of weeks later, because you need to make sure that the mite population is really gone down below that 2% threshold, because especially if you use biopesticides, biopesticides rely on temperature and various other atmospheric conditions, and so their effectiveness is really variable just depending upon when you're using the treatment. So it's always smart to monitor and make sure that the treatment was effective, because otherwise you might have to do something else and you won't even know it. So the sampling methods, I don't really want to go much into sampling methods because I'm going to point you to a reference that will give you all the information you need, including videos. But suffice it to say that the two that are thought to be the most accurate are the powdered sugar shake and the alcohol wash. And both of these are basically taking a sampling of 300 bees. You're taking a half a cup of bees, which works out to about about 300 bees, and you're putting them into either the alcohol or the sugar, and you're shaking them, and then you're basically waiting to see how many mites are going to come out of that sample. They want, I think most of these thresholds for sampling are that you want no more than 6 mites every time you sample. Because 6 mites will correspond to a 2% threshold. So if you get 6 mites or more, you maybe want to do something in terms of reducing your mite loads, just kind of depending upon what time of year. Well, to kind of give you some idea of why it's so important to keep your mite load at 2% or below is because even at 3%, if you have 3 mites per 100 bees, just 1 more mite per 100, that can result in very significant honey losses. There have been all sorts of studies that have, the Canadians, for an example, the Canadians are freaks about Varroa mites, and they monitor and treat really early in the season because the Canadians, they're all about honey production, right? And so they found that if they can keep really low mite counts starting out early, that really, more than anything else, really helps them in terms of their honey production. And of course, at 5%, you got 5 mites per bee, per 100 bees or more, then yeah, your collapse is possible, all because of this mite virus complex. So if you got mites, and you do have mites, don't freak out. There's things you can do. It brings us to, first of all, the cultural tier. You start at the bottom of the pyramid and move up. For cultural controls in terms of Varroa mites, the biggest thing is going to be to manage the queen's genetics. So what does that mean? Well, there's various genetics, pools of genetics that you can purchase that will allow your queen to head colonies that will have behaviors that will naturally keep the mite population low. One of these is the Russian bees. Now, the Russian bees, they've had the longest natural experience with the Varroa mite, but in addition to that, and I think it was about the year 2000, Dr. Rendier brought over a bunch of Russian genetics and basically selected for both honey production and low mite loads. And then he sold them, or didn't sell them, he gave them to the RussianBreeder.org, and that's the email here, or the website here. And so you can go to this website and you can find the breeders that breed these particular sorts of bees. But these bees have been selected in order to groom mites and bite mites and various other things, and they keep mites traditionally around 2%, which is exactly at the action threshold, unless there's immigration or something else that's unusual that's going on. But they are pretty effective in terms of reducing the mite loads. In fact, there's been evidence suggests that you get a 50% efficacy, or I should say 50% effectiveness in reducing your mite loads by the use of these genetics just in general. And then there's the Varroa sensitive hygienic bees. This is kind of interesting. So these particular bees, what they do is they're able to smell through the capping in a cell and tell not only if Varroa mites are there, but also if Varroa mites are reproducing. If Varroa mites are reproducing, then they will actually pull the pupa out, which will then basically dry out the male mite and interrupt the reproductive process. And so this is a behavior that was selected for at the Baton Rouge lab as well. It is really quite effective. They're talking about 50% efficacies on these particular mites as well, or bee lines as well. And all you need to do is just Google Varroa sensitive hygienic bees, and there's all sorts of bee breeders that will sell you bees that have this particular type of behavior. It's kind of funny, years ago, when they started seeing this kind of behavior in colonies that were infected by Varroa mites, they just thought it was a new disease because they would just see bald pupa heads, in their colonies, and they'd never seen that before. But really, that was a good thing. It was the bees that were uncapping the pupa that were infested with reproducing mites, and they were just disrupting that reproductive process. So then there's the live or let die bees. These stock of bees are by bee breeders that really don't care what mechanism the bees are employing to keep the Varroa mite load low. What they're doing is they're just monitoring the mite loads, and if the bees are If the bees are producing a lot of honey and they're relatively docile and they're keeping low mite loads, then they breed from those colonies. And so there's all sorts of mechanisms that the bees are employing, although they're not really quantifiable. We don't really know exactly. But suffice it to say that the bee breeders are convinced that they're keeping low mite loads in those colonies. And so you can just Google survivor stock bees to get bee breeders that will gladly sell you queens that they've selected for with these kind of behaviors. And so there's all sorts of other cultural controls, but I'm not going to go into them because I don't really have the time, but that's really the main one. And remember, you get a 50% reduction in your mite load day and night, all year long, just from that one simple queen purchase. Highly recommended. The second tier of the pyramid is the mechanical controls. And so we're going to talk about some of these. These are mechanisms that you employ while managing your bees to help reduce the number of mites in your boxes. One of the most common mechanical controls is the screen bottom board. And you can see the screen bottom board here. And the idea is that there's going to be a natural small attrition rate of mites falling off bees. And when they fall off the bees, they fall through the screen and can't get back up on the bee. And so effectively you're reducing in a very small way the mite load. Now this is not enough, right? If you just slapped screen bottom boards on the bottom of your boxes and say you're good to go, you don't have to worry about Varroa mites, you would be mistaken. You get a 15% decrease in your mite loads, possibly with this, according to a University of Florida study. But the problem is that that's not nearly enough. But remember, that's what integrated pest management, though, is all about, right? It's not employing one thing, it's employing multiple mechanisms at different angles, each taking a little bite out of the mite population. So in total, in theory, you stay below threshold. Then, of course, there's drone trapping. This is kind of interesting. So you can buy these frames that you can see here. These are referred to as drone frames, and they're always green. In the drone cells, because the drones are male bees, the male bees are a little bit larger, so the cells are larger, right, for the male bees. And the post-capping period for a drone, for our Western honeybee, is 15 days. Now remember, the post-capping period for the worker was 12 days. Now that may not seem like a very big difference, but to a Varroa mite, that's a huge difference, because although in their reproductive cycle within the cell of a worker, they can generate 1, maybe 2 mated females, but they can generate 3 or maybe 4 mated females because of that extra 3 days going from a 12-day post-capping period to a 15-day post-capping period. Because remember, there's a lot more mites in those cells, but only the most mature mites are living and escaping. The immature ones are dying. So the extra 3 days that allows the mite to grow and to get older, develop, is a really big deal in terms of reproduction. Now, because of the selection pressure for mites, they gravitate towards these drone cells because there's been selection pressure that allows their behaviors to understand that it's much better for a foundress mite to jump into a drone cell versus a worker cell because of this extra developmental period. They get a bigger bang for their reproductive cycle in these drone cells. And so effectively, these drone frames are Varroa magnets. And so the idea is that you take a couple of these frames, you put them on the outskirts of the brood area, and you want to do it, of course, in the early spring when they're going to actually, have drones. And the idea is that they, the queen will lay, she lays an egg and she doesn't fertilize it, and so it becomes a male. And the mites smell that, and then the foundress mite will smell that and jump in there and then reproduce. And so these, in theory, should be loaded with Varroa mites. And the idea is that after your cells are capped, your frames are capped, you take them out, put them in the freezer. That, of course, kills the pupa, but it also kills the Varroa mites. And then after you take them out and defrost them, you put them back where you took them from, and the bees will clear out the dead pupa and the dead Varroa mites. The queen will relay, and then you're attracting, again, bunch of Varroa mites to these frames. Now, there's some downsides to this, right? I mean, the upsides are this is completely organic and it is effective. Typically, there was a French study, I think, that said that it was had a 25% efficacy rating in lowering mite loads. But it's laborious, right? If you had lots and lots of colonies, you know, I can tell you the Blake's not going to be doing this. And it's energetically costly for the superorganism to the extent that there's a lot of protein that's being wasted because they're laying these drone pupae and then they're being fed and brought to development. So there's a loss of protein. And the wax is energetically expensive. The wax takes it really takes a lot of honey to produce wax, and so that could have a temporary effect on your honey production. So there are some downsides to it, right? But it is organic and it can be effective and it can be a part of one tool in your integrated pest management tool belt. 25% efficacy, according to a French study. Okay, so then there's mechanical brood disruption. And the idea here is that we know, you know by now that Varroa mites reproduction is directly predicated upon the bee's reproduction, namely the brood area and how many eggs have been laid so that the foundress mites have a place to reproduce. So the idea is that if you can artificially reduce or restrict the amount of brood in a box, then you can artificially reduce the reproductive rate of the Varroa mite temporarily. And so they, you know, like in, there's some countries like Italy, for example, will put a queen in a cage in the summer for a week. And that allows a lot of the brood to clear out, and then it really drops the number of Varroa mites because they don't have a place to reproduce for a while. It's also why almost all commercial beekeepers split. Well, the commercial beekeepers are splitting not just because to make money, but because actually splitting up the colony will temporarily disrupt the egg laying. And so as a result of that, you get some help in terms of your overall mite counts temporarily with this mechanical brood disruption. And so this brings us to biological controls. Now, at least up to this point, although they're studying some things, there are no biological controls for Varroa mites. The biological controls, it's kind of like the concept of buying ladybugs and letting them go in your garden so they eat aphids, right? But we don't have anything that really eats Varroa mites effectively, at least currently. They are working on it. Then you've got the biopesticides. So these are the essential oils and the organic acids and what have you. And that's what I'm going to talk about now. Now, let me start by saying is that you want to start with cultural controls and, like, let's say, for an example, buying a queen that's resistant to Varroa mites, like Russian, for an example. And while testing, if you notice that your Varroa mite loads are going up above 2%, then you could try some of these other things. And then if they're still going up above 2%, then you want to move to the essential oils. In other words, you don't want to jump right to the essential oils and the organic acids. You want to try all these other things first that are less stressful on the bees, which is really the whole point of the integrated pest management pyramid, starting at the base and moving up, but only as it's required to keep your pests under threshold. And so what is it that, what are these biopesticides? Well, these are chemicals that are in nature that the bees are largely pre-adapted to. Like formic acid, for an example, is one of the chemical controls, and there's that naturally occurring in honey. These are all food-grade ingredients, so they're not dangerous to humans. And the main thing is that they kill holistically, so there's no resistance that's created, or at least that's the likelihood. By that I mean is that these biopesticides disrupt the mite, kill the mite in so many different ways that it makes it very difficult for the mite to be able to create a resistance to it. Whereas the synthetic insecticides or miticides, they will kill by disrupting a very specific mode of transfer. And so as a result of that, it's easy for the mites to be able to create some kind of resistance because they're killing not broadly, but very narrowly. And I'll explain more about that in just a moment. And that they are not thought to contaminate wax or honey. These are all products that are made from some of the miticides that are on the market, if you're wondering why I have these things on the bottom here. So let's talk about Apiguard. I like ApiGuard quite a bit. This is really the time of year that you would start trying to use it. It's a gel, it's in a tray. You put it above the brood area. And the idea is that because it's heavier than air, it kind of sinks down, and the bees will try to eliminate it, and it just gets spread around, and it kills the mites. Now, it won't kill the mite underneath the cells, but it kills the mites that are on the outside of the bee. There's a 93% efficacy on this generally, which that's really, really high. It's a thermal product. It does kill some brood. You can't put the supers on with this stuff. You got to have supers off. You can keep the supers on and use it, but your honey might give you kind of a spearmint-y sort of breath, later on in the year, which is not what you're going for, because it's what they make Listerine out of. So you don't want to have your honey supers on. Also note that they indicate using a half a dose if the brood temperature or if the outside ambient temperature is at 77 degrees as a high or higher. In other words, if your daytime temperature is going to hit 77 or higher, you want to use a half a dose and not a full dose. I had a friend who used a full dose when it got hot out, and that's why you have all this bearding out here. They eventually all went back in, but it did disrupt the colony and it was unnecessary. It's much better to follow the directions. You apply it 2 times, 14 days apart. And remember to use it with a daytime temperature of no higher than 100 degrees Fahrenheit. The other biopesticide is the Mitaway Quick Strips, the formic acid. It comes sort of in pads like this. You can use 1 pad or 2 pads. 2 pads is a lot more effective than a single pad, but of course, this is probably the most stressful of the biopesticides. So you only want to use it, and you only want to use 2 pads if you really need to use it. And that's why it's much higher on the integrated pest management pyramid, because all these things will cause a certain amount of stress. I mean, these are things that you don't want to use unless you have to use them, but they just beat the heck out of your colony succumbing to Varroa mites. And so this is what you have to do in order to keep them under control, and you just need to do it. So let me kind of give you some more specifics on this. It is stressful. It can kill some brood, but the research seems to indicate that although it kills some brood, the queen will go up into the box above the treatment and wait for the treatment to dispel about a week later, and then come back down and she'll lay like a son of a gun. So my understanding is there's no long-term decrease in the amount of your adult population for the use of this product because the queen will catch up. This can be used with supers on. I mean, that's great. That's amazing. And you can use the knockout or the knockback option. Just by that I mean you can use 2 pads if you want to get up in the 90% or higher efficacy, or 1 pad to get like 50 to 60% efficacy. So I would use, ideally what you do is you monitor your mite loads so that right before, just as you're approaching 2% or get to 2%, you just use 1 of the pads, right? If you waited because you weren't monitoring your mite loads and then your mites got really, really high, say 10%, well, man, you're in critical area. At that point, you're going to have to use those 2 pads because you're right on the edge of collapse if it's not already too late. So the big downside to this stuff is that you can only use it up to maybe 85 degrees in temperature Fahrenheit. But you only use 1 treatment, so it's really nice. You just put 1 and you forget about it. Go back later and throw it away. It is very effective, 60 to 95% efficacy. Okay, so oxalic acid. This was recently, several years ago, it was approved for the first time in the United States. It exists, this acid exists in spinach. We eat it all the time. So it's not bad for humans. Used to be Brushy Creek had it. Now they were out of business, but you can buy it at any of the other bee manufacturing companies. And basically, you can use it 2 ways. You can use it as a fumigant, or you can use it as a dribble. Now, I don't use the fumigant. There's a lot of people that swear by it, but the dribble is easy enough for me, and I don't have to worry about getting a mask, a respirator, or any of that problem. So I just use this if I were to use it. And then you just mix this stuff up with sugar water. And you want to use water that's soft. So don't want to use hard water because for some reason it makes it ineffective. So you need soft water. It needs to be, you need to mix it with sugar syrup. And there's directions for all of that. And I'll direct you to the proper resource. The thing is that you want to use it. It kills the mite on contact. So it's not going to kill mite underneath the brood, but it will kill the mites that are on the bees. And it's a flash kill. In other words, it kills all within like 30, 45 minutes, that sort of thing. There's not a lingering kill. And so because it's a flash kill and not a lingering kill, you're going to have to use it when there's no brood. There's been all sorts of studies that have indicated that even if you have a brood that's the size of the palm of your hand, you just had that much brood, that would hide enough of the Varroa mites to make a lot of this ineffective. Because remember, not in the winter, but in normal times, the mites are going to be, most of your mites are going to be underneath the capping in the reproductive stage. And so things that kill mites on contact are not going to be really effective. It's a flash treatment unless you have little to no brood, really no brood. So what they're telling us now is that if you have just in the winter, like in December, if you just had like a little bit of brood, maybe the size of your hand, that you would just actually take a fork or something and just kill all that brood so that the chemical will kill all the mites that could be underneath the capping in that case. They're not sure what the mechanism is, why it's killing the mites, but they think it's being, the oxalic acid is being absorbed through the dorsal claws of the mite. Now, I don't expect you to know how to use this product or any of these products on the basis of this presentation. It's going to be important for you to, I'm going to direct you to a resource that's going to go into great detail in terms of how to use all of these and when to use them and what you should use and what you shouldn't use. So oxalic acid is not lipophilic. In other words, it's not absorbed by the wax. You can use it, it has a broad temperature ratio, but or window, but you can only really use it in the winter if you're going to use the dribble, because it's you got to look for a time where there's just really no brood for it to be effective. But it's really easy, it's inexpensive, it's not very caustic to the bees. In other words, it's probably one of the least stressful abatements used. And it's highly effective. They have up to a 97% efficacy with this product. So that's the biopesticides. Now this is where I stop. But the truth is, integrated pest management is you don't go any higher on the pyramid than you need to keep your mite load below action threshold, right? Which is 2%. And I'm able to, I mean, anybody can really. Randy Oliver, he's got a commercial operation and all he uses is biopesticides. So if you're a hobbyist or somebody with 20 or 30 colonies or less, then biopesticides shouldn't be a problem at all for you to be able to effectively control your Varroa mite loads. So I don't go into the synthetic miticides in regards to my personal use, but let me tell you, they are a legitimate part of the integrated pest management system, and it beat the heck out of all your colonies dying in case the things you're using haven't worked up to this point. Now, let me tell you, and I'm sure Blake would agree with this, of the 3 miticides, the one to use is, if you're going to use a synthetic, would be Apivar with Amitraz. This does have some downsides to it, but it's not nearly as toxic as these other chemicals, the Checkmate Plus and the Apistan. In other words, I can't believe these things are still on the market. They contaminate the wax, you know, these chemicals up here, Apistan and Checkmate, the wax actually captures the active ingredient and causes all sorts of brood hardship and death potentially for years to come. And they also mix with other chemicals and create negative synergies that increase the toxic effects of these chemicals in your wax. So really, this is going to be by far the best chemical to use. In addition to that, these other ones are largely, the mites have tolerated them. There's tolerance to it. They're not even very effective anymore. But if you're going to use a synthetic, this is the one to use, but there are some downsides to it. There's no doubt about it in regards to reducing the egg. I've heard that it reduces the egg laying rate of queens to some extent. There was a Walsh study in 2014. Coumaphos and fluvalinate, which were the first 2 chemicals for sale, they lower sperm viability. All of these things contaminate wax and honey except for the biopesticides. There's all sorts of nasty synergistic effects that can be created through these use of synthetic chemicals. But they have their place, and especially if you're a commercial beekeeper, you don't have a lot of time to effectively use a lot of inputs. And so for commercial people, it's necessary to use, to maybe use chemicals. You know, there's no judgment here. But I think that if you're a hobbyist or something like that, you should have no trouble whatsoever using biopesticides effectively and keeping your mite counts lower. So, Mark wants to know, when should you treat? Well, that's going to be, that's really debatable, right? But to use this graph that we had used earlier to kind of give you some idea, January through December, I would use mite away quick strips, or I do, in February or early March, because remember, they're effective from a temperature range of 50 degrees to a high temperature of 85 degrees. And so you're able to get a good effective kill, and it's not so hot that too much of this product gets released in your colony. So it's not very damaging whatsoever, and does a really nice number on the mites. Okay, well, so then you're putting the supers on. Now you could use mite away quick strips with the supers on in case you needed to control your mite level at this point, except May, June, and July is going to be too hot, because remember, the window only allows for a daytime temperature of a max of 85 degrees, right? So what do you use? Well, Apiguard. You can use Apiguard up to 100 degrees. Now in Dallas, I looked it up on weather.com. This week is too hot, but next week, according to weather.com, your daytime temperatures are going to be between 92 and 94 degrees as a high. That's a week away, and of course we know about weather forecasts. They're not always very accurate, but you might keep your eye on that. This is a critical time to use Apiguard, because you can see how the mite population could potentially be ready to skyrocket because of this exponential curve. And so it's necessary to get a knockback treatment so you can stay below 2%, so that you've got clean winter bees going into the winter, right? We want a really low mite count right before your winter bees are being made, so that you've got clean bees that will likely have long longevity to last, all the way through the winter. And then oxalic acid. You could use oxalic acid if you want to do a cleanup in December with very little brood. You use the dribble, and that'll keep you really clean going through January and February, and maybe even to March. So this is the takeaway. You got to know your mite levels, because if you don't know your mite levels, you're just flying in the dark. And if you wait till you see a mite, then it's just really too late. Your virus counts, the viruses are just out of control, and they're just careening throughout your colony. It's very difficult to get that genie back into the bottle. One of the easiest ways as a basis of having low mite counts is just to use mite-resistant stock, right? These are bees that just naturally help clean out the Varroa mites in the colonies, and it's completely organic. Now, if that's not keeping you under that 2% action threshold, then you might want to use a combination of these biotechnical strategies before you escalate, like the screen bottom boards, for an example, or drone trapping. And then if your mite counts are growing, then you need to move to the biopesticides. Now remember, biopesticides, they're organic and they're sustainable, but they are stressful for the bees, so you only want to use them if you need to. But goodness sake, if you need to, and you need to use them, because they are extremely effective. And the Varroa mite, there's not much that will kill a colony quicker than Varroa mite infestation. So let me just leave you with this. You got to be proactive, right? The thing is that you can screw up all sorts of things as a beekeeper, and the bees will forgive you. But not managing Varroa mite levels is not one of them. That is the dragon that you're going to have to slay as a beekeeper if you're going to be effective long-term in keeping healthy colonies in your apiary. So be proactive. So as promised, this is the resource. And of course, you guys, your club was talking about it already. Honeybee Health Coalition, they've got a manual that goes through everything and more that I've talked about today, and lots and lots of detail, and also lots and lots of videos. So it kind of helps explain how to use the various products and when to use them. And so this is really, this is the latest and greatest information from the bee scientists. So I really can't stress it enough that that's really what you ought to do is look it up on the website and just get going on it. I think that you'll learn a lot. And so that's it. That's all I got. I guess we need to see if there's any questions.
Yeah, hey Lance, I'm tracking the questions for you, but what do you have on as far as studies for the thermal treatments?
Okay, so the thermal treatments, I don't know of any specific treatment, any specific studies on it, but let me tell you what I do know. Dewey Caron, he's an apiary scientist, super nice guy, and he's done countless amounts of studies on Varroa mites and any number of other things. And he says his belief is that thermal treatments can be very effective. And so the thermal treatments, in case you guys don't know, it's this concept where basically mites will die at 106 degrees Fahrenheit. Bees will die at like 112 degrees Fahrenheit. So the idea is that you put this device in your colony and it ratchets up the temperature, the ambient temperature within the colony to 106 degrees, killing the mites and in theory, not killing the bees. And I think that they ask you to keep this thing going for maybe 2 hours or so before you move it on to another colony. So the advantages of this is, yes, it's effective, probably. And the other advantage is that it's organic. The disadvantage potentially is that we don't know how that could affect the queens. Jeff Pettis from the USDA lab, he did some studies that indicated that if queens were subjected to higher temperatures, it could effectively kill the sperm, or some of the sperm in the spermatheca. And that results then in premature supersedure, right? Because your queen starts laying a bunch of drones, and then the colony realizes that and decides to replace that queen. So I'm not saying that that does happen. I'm just saying that there is a educated risk that could happen. I talked to David Tarpey about it. It was one of the, probably the premier queen, well, one of the premier queen experts, along with Dr. Rangel. And what he said is that they don't know, it could affect a queen in a negative way, but they just don't know. But they do know that it kills mites. So I'll just leave that up to your own discretion to decide what you guys want to do. Any other questions?
I had there's one here. It says, upon what recipe or mite load is the recommendation for the 1.5 cc per stream of the oxalic acid?
What's the first part of that question?
It says, what recipe or the mite load?
Yeah, you're going to have to go into the resource that I put up. Go ahead and look that up, because it's going to be everything you need to know in terms of how to mix that and when to use it and how to use it. All of that's going to be in that Honeybee Health Coalition material. Also, I would recommend that you Google Randy Oliver. Randy Oliver, he's a national treasure, in my opinion. He's a scientist, a bee scientist, and a commercial beekeeper. And so he's always doing a lot of studies that are really pragmatic and making us better beekeepers. And he's magnanimous, he just shares all this wonderful information to everybody who wants to use it. But if you Google him, he also has all sorts of YouTube videos and information on his website in regards to how to use oxalic acid in terms of the dribble.
All right. And then another question here says, are you recommending Max Apiguard and OA to be used in succession throughout the year or as a standalone treatment once per year?
Well, that's a good question, but the truth is it depends on your apiary, right? So all apiaries are different. So some apiaries, for an example, maybe you don't need a treatment. Maybe you just have, you employ Russian bees and that's going to be enough. Other apiaries, maybe you just need one treatment a year, depending upon, you're monitoring your mite loads throughout the year. You should at least monitor them 4 times a year. And so the question cannot really be answered definitively. You've got to monitor your mite loads and decide if your area, if there's so much of if there's so many mites in your particular area that results in you exceeding threshold pretty quickly or often, you're going to have to use more than one. Typically, I think, at least in my area, I use 2 or 3 a year. Every year is different. But let me go back to my original point. Every area is different because what they're learning is that most of the pressure from Varroa mites is not necessarily the internal reproductive rate of the mite, but rather the fact that other colonies within a half a mile radius of your colony are collapsing because the beekeeper is not monitoring their mite loads, or maybe it's a feral colony. And then that honey, when that colony depopulates, gets robbed out and brings all those mites to you, right? So, so there's that kind of environmental pressure that's different from one area to another, just depending upon how many feral colonies you have or how many beekeepers are in your area. The other thing that's different from area to area is the actual cocktail of viruses. Some locales will have viruses where the viruses are not nearly as virulent or harmful to a bee colony as others. And so it's a combination of those two things, right? The density of colonies and consequently the density of the Varroa mite populations that's adjacent to your colony, and what's the ambient virulence of the viral loads in that particular area. And each area is different and it changes. And so there's no way to know that, to know those things so that you can somehow manage to compute how many treatments you're going to need in advance. The only thing you can do is test your mite loads and see how much pressure there are from this parasite before you decide how many treatments you're going to ultimately need in a year.
Another question was, is if the colony is showing viral stress now, is it too late to try a treatment, requeen, or combine a hive?
Well, well, first of all, you don't want, if you've got Varroa mites in your colony, the last thing you want to do is combine it with a healthy colony, because you'll crash that colony. So what you want to do is that because you don't know how damaging the particular cocktail of viruses is, that even though you've exceeded your 2 mites per 100, that's not to say you should give up. No, by all means, even if you've got excessively high mite loads, use a treatment. If you've got really high mite loads, though, you're going to have to use a biopesticide. That's going to be the only thing that's going to be as fast and effective to crater your mite population quick enough, because you got to do something quick. So yes, what you want to do is you definitely want to use a treatment, and you want to try to save your colony. You definitely want to use a treatment if for no other reason but to keep other bees from rotting out your colony that's going to ultimately collapse with all those Varroa mites. You don't want those Varroa mites spread. In fact, if you couldn't treat your colony, what you want to do is you want to just tape it up and let the bees die, because, golly, the Varroa mites in the colony is poisonous to your other colonies. But the best thing to do, of course, would be to treat it and crater those mite loads and hope that the viral cocktails in your particular area are not so damaging that mite load is going to ultimately result in the demise of your colony. There's really no way to know that for sure. So I would just treat the best you can and hope for the best. The main thing, the moral of the story, is to hopefully not be able to get to that point where you're having to make that those very depressing judgments. Hopefully you're monitoring enough and getting a feel for the mite population dynamics in your apiary, so that you know how often to treat, and you'll start learning when to treat, and hopefully you won't have to make very many of those really terrible decisions in regards to having a really super high mite load in a colony.
Yeah, I think this is the last question. Was somebody saw something about the new robot on the news and want to know would it be effective on the virus? I haven't seen anything.
So, there's a new robot on the news?
That's what somebody mentioned. This is the new robot that they saw on the news would have an effect on the virus.
Yeah, no, I'm sorry, I'm not familiar with that.
Yeah, I haven't seen anything either.
Robot sounds good to me, man.
All right, Russell.
I have a question.
Okay.
How aggressive is the temperament of the Russian bee?
You know what? It's so fascinating that every time I talk about Russian bees, everybody has that question. And I think I know why people have that question. To answer your question, Russian bees are notorious for being relatively docile. And I it true in my apiary as well. You know, I keep an apiary of feral bees and an apiary of these managed Russians. And the difference in temperament is night and day. It's just unbelievably different. They're so pleasant to deal with. Now, to go back to my original point, the reason why I think people are always asking me that question is because it is known, it's widely understood, that when Russians cross, in other words, if your Russian queen that you bought dies and they make a queen, they send it out to the drone congregation area in your area, you have, at that point then, what you're going to have is a cross of a Russian and whatever cocktail of genetics is out there in your area, right? So you're going to have a hybrid bee. Now, for some reason, these crosses have a tendency to be, at least on some occasions, aggressive. And so I think that what happens is that people buy Russian bees, and a few months later, their colony gets superseded, and that was invisible to them. They didn't notice, and all of a sudden, their colony is real defensive, and they say, "Oh, it's the Russian bees. I guess they're defensive." When really, it was just that you've probably got a cross. You don't have a pure Russian line. So yes, they are very docile, with a few exceptions, but I would requeen them every year because you want to keep that Russian genetics from crossing if you're worried about them being aggressive.
And the person that posted about the robot said it was supposed to be used to kill coronavirus in a building. So
Okay.
I don't know if we've got anything that small for beehives yet, but
Yeah, not to my knowledge.
You're turning red. Okay.
Lance is trying to keep a straight face.
I've been asked more unusual questions than that, believe me.
All right.
Good job, Lance.
Okay, well, good. I hope everybody learned a little something that will be helpful.
Well, I've
yeah, we always appreciate it because I know you're there during the summer clinic and the TVA, and it just seemed, since everything's being shut down, that this is an important thing out there, and you're pretty well versed in all the different IPM methods. You know, like I said, with the commercial side, we tend to lean towards that synthetic chemical for our late honey or the July kills, and then use the others. I've been using oxalic and formic and trying some other stuff for my cooler weather kills. So it has been effective so far. So I'm hoping to have some better results with some bio-organic stuff instead of going synthetics.
Okay, great. You got to do what you got to do, you know. The commercial beekeeping is so difficult, man. You guys are the hardest working people on the planet.
Or crazy.
And biopesticides are much more difficult to use than synthetics. So I certainly understand. We got to keep the bees alive so that we have food to eat. But I think really that if you don't have a huge number of hives, I think that especially hobbyists, I think you guys will find that you can be very effective in just going up to the biopesticide level.
Thank you,
All right,
Lance.
Russell.
All right. Well, again, Lance, great job. Appreciate you coming on. And
Hope to see you soon.
yeah.
I hope to see everybody soon. Well, I really enjoyed it. If you guys have any questions, you can always email me, lance@beekeepinghelp.com.
Thank you.
Okay, thanks, guys.
All right.
Bye.
Thanks. Good night.
Bye.
All right. If everybody hasn't gotten off, just give you a heads up that Collin College has shut down through January. So we will be doing Zoom meetings through January for sure. That will be the end of tonight's show.